Hi, I am Dr. Arsalan and today we are going to solve the MCQs on lipids. The first question is, oils are dash fatty acids. Given options are saturated, unsaturated, natural or none of these. As we know that the lipids are made up of fatty acids. There are two types of fatty acids, the saturated fatty acids and unsaturated fatty acids. Saturated fatty acids are those fatty acids in which there is single carbon-to-carbon bonding. These are solids at room temperature like fats. While the unsaturated fatty acids are those fatty acids in which there are multiple bonding between the carbon atoms. There may be double bond or triple bonds. But the condition is that there must be at least one double bond between the carbon atoms. And these are liquids at room temperature, for example, the oils. So he is asking about the oils. So oils are unsaturated fatty acids. Option B is correct. The second question is, phosphate head of the phospholipid is polar hydrophobic, polar hydrophilic, both are none. So that the phospholipids are those kind of lipids in which the phosphate group is attached to the fatty acids and glycerols. So phospholipid is made up of three components: the glycerol, fatty acid and phosphate group. Phosphate group act as the head of the phospholipid. The phospholipid, the phosphate, and it is polar, it is negatively charged, and it is hydrophilic, that it attracts the water molecules. Option number 2 is correct, that the phosphate head of the phospholipid is polar as well as hydrophilic. Next question is, high content of triglycerides is found in. Given options are low density lipoproteins, high density lipoproteins, chylomicrons, or very low density lipoproteins. To solve these MCQs, you should know about the triglycerides. Triglycerides are basically the saturated fatty acids. These are also referred to as animal fats. Option low density lipoprotein and high density lipoproteins both are made up of fatty acids and cholesterols in the liver. But the chylomicrons are the large molecule of triglycerides. These are made up of lipoproteins and are synthesized in the enterocytes, the intestinal cells, which synthesize these chylomicrons from the dietary lipids. And these are made up of cholesterol, fatty acids and glycerols. So the option chylomicron is correct, that these are the large molecules of triglyceride having high content of the triglycerides. Next question is, for determination of the fatty acids composition, the process of esterification is implied in which aqueous solution is used. He is asking about that the fatty acid composition. This question is very informative because it is giving you another clue that the fatty acid composition in the lipids can be determined through esterification. For esterification of the lipids, a warm solution of what kind of compounds or molecules is necessary or required in these reactions. So the given options are methanol and sodium hydroxide, methanol and potassium hydroxide, ethanol and hydrochloric acid, ethanol and bicarbonate. So it should be kept in mind that if for esterification of the lipids, for determination of the composition of the fatty acids, two kind of solutions can be used: methanol and hydrochloric acid, or methanol and sodium hydroxide. So the given option A is correct, methanol and sodium hydroxide. This is the correct option here, which can be used for the determination of the fatty acids. Next question is, the fatty acids are dash in nature. He is asking about the nature of the fatty acids, whether these are charged or non-charged molecules. For example, given options are polar molecules, non-polar molecules, amphipathic or neutral. Amphipathic means that having both polar and non-polar ends. So the lipids or the fatty acids have both polar and non-polar ends. For example, in phospholipid, we have seen that phosphate end of the lipids, it is polar, it is negatively charged, while the rest of the fatty acid was non-polar, having no charge. So the amphipathic option is suitable for this because the lipids or the fatty acids bear both polar as well as non-polar ends. So the option C is correct, amphipathic only. So fatty acids are amphipathic in nature. Next question is, the triglycerides are hydrolyzed by alkali treatment with potassium hydroxide or sodium hydroxide. The process is called. This is again an informative question and giving an information that triglycerides are hydrolyzed by alkali treatment. So these can be hydrolyzed by the addition of alkalis like sodium hydroxide or potassium hydroxide. So this should be kept in mind that these two alkalis are used to hydrolyze the triglycerides. However, the process is called, given options are alkalization, neutralization, hybridization or saponification. Alkalization is the addition of alkalis, neutralization is the addition of alkalis to the acids, hybridization is the mixing up of the two chemicals. Saponification, this is the technical process of formation of soap. Saponification is the soap formation. In case of soap formation, the triglycerides are used and the alkalis are added to these triglycerides to hydrolyze these triglycerides, and then soaps are formed. So this process, basically the triglycerides in which alkalis are added, the sodium hydroxide and potassium hydroxide, is the formation of soap. So this is called the saponification. The option D is correct. This saponification or soap formation. Next question is HDL, the high density lipoproteins are synthesized in dash. Given options are blood, bone marrow, liver or intestines. So this is fact based question, and it should be kept in mind that the high density lipoproteins as well as low density lipoproteins both are synthesized in the liver, while chylomicrons or triglycerides are synthesized in the intestines. So the HDL and LDL are synthesized in the liver. Next question is the specific gravity of lipids is. Given options are 0.5, 0.7, 0.8 or. This is again fact based question, and it should be learned by heart that the specific gravity of lipids is 0.8, because lipids are lighter than water and they float on the surface of water. Their specific gravity of water is 1, so their specific gravity should be less than 1 because they are floating on the surface of water and they are lighter from the water molecules. Next question is dash cell in invertebrates store fats. Given options are hepatocytes, enterocytes, adipocytes and chylomicron. We have learned in this lecture about the cells that the hepatocytes are the liver cells synthesize the high density lipoproteins as well as low density lipoproteins. The enterocytes synthesize the chylomicrons. The chylomicrons are the molecules, large molecule of triglycerides. These are the molecules, not cells. Adipocytes are the fat storage cells in both animals and human beings. These store fats in the body. So this is the correct option, the adipocytes. Next question is dash emulsify the fats in the intestines. Given options are bile, pancreatic juice, lipase and amylase. Basically, fats are emulsified in the intestines. Emulsification is the process in which fats or lipids are surfaced upon the chyle. The chyme and chyle are the pre-digested material in the intestines, and the lipids which are dissolved in this chyme or chyle are brought to the surface of the chyme or chyle for the action of lipase for further digestion process. So this process in which the lipids are surfaced or brought to the surface of the chyle, this is called emulsification. The first option, bile, is correct because bile comes from the liver and gallbladder through bile duct, common bile duct, and this bile is required for the emulsification of fat. So the option A is correct. Rest of the options, pancreatic juice, it comes from the pancreas through pancreatic duct and has multiple enzymes in it, lipase, amylase, amylopectin, etc. There is no concern with the emulsification of fats. The lipase digests the lipids and the amylase digests the carbohydrates. So these are the enzymes. The bile is basically the salt solution which aim to emulsify the fats, surface the fats on the chyle for the disposition of the action of lipase. Next question is asking about the normal cholesterol values of the blood. So the question is normal total blood cholesterol level is. About the total blood cholesterol, for example HDL, LDL, VLDL, all of the cholesterols are connected in this. So given options are 1 to 2 grams per deciliter, 5 grams per deciliter, 10 to 20 milligram per deciliters and 100 to 200 milligram per deciliter. This is fact based question, and it should be kept in mind that 100 and 200 milligram per deciliter of the blood is normal blood cholesterol level, and it should not be exceed from the 100 or 200 milligram per deciliter of the body. The last and most important question is asking about the kind of lipid. There are two kinds of lipid, the storage lipids as well as the structural lipids. So he is asking that which of the following is storage lipid, which stores energy, which stores fatty acid for further use, or which is the energy reserve of the body. So given options are glycolipids, waxes, phospholipids or acylglycerols. Glycolipids still components of the cell membrane of the cell. Waxes external coating of the insects and plants to prevent evaporation. So these both are structural lipids making the structure of the body. Again, the cell membrane of the animal as a plant cell, it is made up of phospholipid by also a structural lipid. Finally, acylglycerols, it contains triglycerides, fats and oils. So this is the storage lipid. This is the energy reserve of the body. So the option D is correct, that acylglycerols are the storage lipids, while rest of these are the structural lipids. Hope this video will help you a lot in preparing for your medical entrance test examination. And don't forget to mention your marks in the comments box, that how many MCQs you were able to solve and how many MCQs you have learnt in this lecture. Thank you.